Hello students, welcome to Ban Lectures. Today we'll talk about fatty acid oxidation. So this video would be suitable for medical, dental, pharmacy, nursing students and even for students studying PG in biochemistry. So let us see what is fatty acid oxidation and how it takes place in the body. Now, fatty acids, these are organic compounds that fall under the category of lipids. Or we can say fatty acids are supposed to be backbone of lipids. Upon degradation of any lipid, what we get is fatty acids. So lipid cannot exist without fatty acids. So fatty acids can be grouped into two classes, saturated and unsaturated, on the basis of presence or absence of double bond between two carbon atoms. Saturated of course means absence of double bond in the structure, and unsaturated means presence of double bond between two carbon atoms in the overall structure. Now, unsaturated fatty acids can be further subclassified into monounsaturated fatty acid and polyunsaturated fatty acids. Monounsaturated means presence of one double bond. Poly means it can have more than one double bond. Here is the example of saturated fatty acids, absence of double bond you can see, and here you can see unsaturated fatty acids, presence of one particular double bond. Now, fatty acids can also be grouped into three subclasses on the basis of number of carbon atoms in the overall hydrocarbon chain. So we have short chain fatty acids, so the number of carbon atoms will be 6 or less. Intermediate or medium chain fatty acids, number of carbon atoms will be 6 to 12. And finally, long chain fatty acids which bear 12 carbon atoms or more. Now what happens is fatty acids upon degradation, what they give is acetyl coenzyme A. So triglycerols, which are the simple lipids, they bear fats and oils. Upon degradation they would release fatty acids. So fatty acids upon degradation or oxidation or breakdown, they release acetyl coenzyme A. So one source of acetyl coenzyme A is oxidation of fatty acids, that is from lipid metabolism. Secondly, we can also get acetyl coenzyme A from degradation of carbohydrates, that is glycolysis, etc. Thirdly, we can also get acetyl coenzyme A from degradation or oxidation or catabolism of amino acids. So acetyl coenzyme is obtained from three sources: from lipid metabolism, from carbohydrate metabolism, and finally from protein metabolism. Now this acetyl coenzyme A would enter into TCA cycle, then only complete oxidation of carbohydrates can take place. Now, if acetyl coenzyme A is produced in excess under certain clinical conditions, then it would form ketone bodies. And acetyl coenzyme has other applications also, used in the synthesis of cholesterol, and finally cholesterol is also utilized in the synthesis of so many steroids. So this is the background regarding acetyl coenzyme A. Now talking about oxidation of fatty acids, we take example of a typical fatty acid, say palmitic acid. It is a saturated fatty acid. It bears 16 carbon atoms. So upon oxidation, it gives 2380 kilocalorie per mole. Now the mechanism of fatty acid oxidation was studied by Franz Knoop in 1904, known as Knoop hypothesis. So what he concluded from his experimentation, that fatty acids upon degradation, they get degraded or oxidized at a particular carbon atom known as beta carbon as per Greek nomenclature. Fatty acids upon degradation, what they give is they are shortened by two carbon units, and these two carbon units represent acetic acid. So here we can see this fatty acid over here. As per Greek nomenclature, the first carbon atom, this would be alpha, this would be beta, gamma, delta, and finally omega. So oxidation of this particular fatty acid takes place at beta carbon. That is why it is called beta oxidation. Now, fatty acid oxidation, as we said, beta oxidation, that mainly takes place in the mitochondria. That is the primary site for beta oxidation. Now, oxidation of fatty acids can also take place at alpha carbon and also, so we will call it alpha oxidation. So, peroxisome is the primary site for alpha as well as beta oxidation. It handles very long chain fatty acids and branched fatty acids. And lastly, endoplasmic reticulum, a very important cell organelle, is the site for omega oxidation of fatty acids. So omega oxidation means fatty oxidation takes place at omega carbon of fatty acids. Now, what happens in fatty acid oxidations? It is complete in certain stages. Stage 1 is, first of all, we need to activate the fatty acid. The fatty acid which has to undergo fatty acid oxidation needs to be activated, and later on it is to be transported inside the mitochondria. So in order for activation of fatty acid to occur, the fatty acids, after getting mobilized from adipose tissue etc., they have to undergo activation before entering the mitochondria. So this very step is being catalyzed by fatty acyl coenzyme synthase enzymes, which is responsible for activation of fatty acids and couples them with coenzyme A. Of course, this reaction needs ATP. Hydrolysis of ATP molecule takes place, resulting in the formation of fatty acyl coenzyme A molecule. Now later on, these long chain fatty acid molecules are to be transported into the mitochondria, where beta oxidation is supposed to occur. So long chain fatty acids or acyl coenzyme cannot enter into the mitochondria. They need a transport mechanism. They need a carrier. So they are converted to acyl carnitine. Carnitine provides as a transporter, as a carrier to carry these long chain acyl coenzyme A molecules into the mitochondria. So they form acyl carnitine under the enzyme carnitine palmitoyl transferase first, CPT. It is located in the outer mitochondrial membrane. Later on, acyl carnitine chain is transported across the mitochondrial membrane by an enzyme carnitine translocase. Inside the mitochondria, carnitine palmitoyl transferase 2 is also present that reconverts acyl carnitine back to acyl carnitine. Because ultimately carnitine is to be released so that it can carry next molecule of acyl coenzyme A inside the mitochondria. So you can see this figure. This is cytoplasm. Here you have fatty acids. Fatty acids get released from adipose tissue mobilization or triacylglycerols. They combine with carnitine is the carrier. So under the influence of enzyme acyl coenzyme A synthetase. So here you have this carnitine palmitoyl transferase first located in the outer mitochondrial membrane that converts it into acyl carnitine. Acyl carnitine passes the intermembrane space of the mitochondria, enters into inner membrane of mitochondria where you have carnitine palmitoyl transferase 2 that reconverts into a carnitine, and carnitine flows back to the cytosol to carry next molecule of fatty acyl coenzyme A. So carnitine CPT means carnitine palmitoyl transferase. Now, carnitine is basically synthesized within the liver. It is synthesized from lysine, and to carry out carnitine biosynthesis, it needs vitamin C also. Now this is the first step of beta oxidation. Fatty acyl coenzyme A under the influence of acyl coenzyme A dehydrogenase. So this is the oxidation reaction taking place here. FAD is the coenzyme here, gets converted to FADH2. Later on, hydration reaction takes place. So enzyme is enoyl coenzyme A hydratase. So in the first reaction, this dehydrogenation reaction, this hydrogen here a double bond is created over here. As you see in this structure, removal of hydrogen atoms takes place here. Now this hydration reaction, H2O is added here. So this is an addition reaction. So water would be added somewhere here at this double bond. So double bond would get converted into single bond. The product obtained is hydroxyacyl coenzyme A. Again oxidation reaction takes place under the influence of a dehydrogenase enzyme. NAD is the cofactor, we get NADH. Finally, we get beta ketoacyl coenzyme A. Now it is we have to break it. Now under the influence of enzyme beta ketothiolase, thiolysis takes place. We get fatty acyl coenzyme A and acetyl coenzyme A. So we started with fatty acid, palmitic acid. It is shortened by 2 carbon units, that is released in the form of acetyl coenzyme A, and next cycle would start. So this is oxidation reaction, finally hydration reaction, oxidation reaction, thiolysis reaction, as we said just now in the previous slide. Now the FADH2 which we got in the fatty acid oxidation, NADH which we got in the fatty acid oxidation, they would enter into TCA cycle via electron transport chain to generate ATP. So finally, we see the yield of ATP or the energetics. Now, the amount of ATP synthesized from single round of fatty acid depends on the size of fatty acid. Size means number of carbon atoms. For example, we started with palmitic acid. It has 16 carbon atoms, saturated fatty acid. It will undergo 8 cycles, so it will remove 8 acetyl coenzyme A molecules, repeating it 7 times. So it will carry out 7 times. 7 into 2 means 14. So 8 molecules of acetyl coenzyme are released. So accordingly we get 7 mole of NADH, 7 mole of FADH2. So this is the energetics over 7 mole of FADH2 will release 14 ATP, 7 mole of NADH will release 21 ATP, and 8 mole of acetyl coenzyme A oxidize. So from one cycle of ATP we get TCA cycle we get 12 ATPs. 12 into 8 means 96 ATPs. So overall it comes to 129 ATPs. So initially 2 ATPs are utilized for the breakdown of that activation of fatty acids. So the overall energetics is we get 129 ATPs from oxidation of fatty acid like palmitic acid molecule via fatty acid oxidation taking place at beta carbon atom. This is beta oxidation. Thank you.